As एमएन क्लब की मीटिंग्स की दिमाग
तो अभी तक है अभी
अरे एमएन तो ये पर बाहर मेरे को क्या पढ़ाई में की अच्छी नहीं हुई है तो अब आदत है 10 बजे तो लाइक में आओ पर आपसे कॉलेज में facet to अक्षरा Maybe history. Good evening. What did we complete the other day? Tell me.
Animal cell, I think we did. Yep. We need to start with plants.
Animal cells already done.
Yep.
Also organelles we have finished.
Yes.
Mitochondria we finished.
No, I think we had to continue from a particular one.
Which are the organelles you checked that we have finished or not? Huh? Nucleus did we finish?
Yes.
Endoplasmic reticulum we finished.
Yes, ma'am.
Mitochondria we finished.
Yes, ma'am. No, we were on mitochondria.
Check karo. Mitochondria we haven't finished. We haven't finished. Then lysosomes we finished.
We just did two, I think. Yes, ma'am. We were on the third one. We just did cell membrane and mitochondria.
Okay, only these two we studied. So, what I will be studying now is that when we talk about mitochondria is one of the major cell organelle. What did we study? Which was that organelle that we completed discussing?
Cell membrane.
Cell membrane. What was the composition of cell membrane we understood? Composition meaning what is the cell membrane made up of? What is the cell membrane made up of? Lipoproteins. What is the meaning of lipoproteins? Proteins and lipids. So when I talk about proteins and lipids,
I'm feeling all on the family.
oh, sure. Both or one only? So when we talk about the cell, diagram drawing you have to practice. Did you try drawing the diagram again?
Yes.
I did try.
The answer.
See, you'll have to practice the diagrams, plant and animal cells both. So we had finished with the nucleus, suppose. Now when we are talking about the structure of the nucleus is something like this. We finished discussing the nucleus, did you say that?
We did choose
Only cell membrane, cell wall you are saying?
no cell membrane and mitochondria.
Okay, cell wall also we haven't finished. So now in that case, let us look into mitochondria. How would the mitochondria look like? This is the basic structure of the mitochondria. What are the features? So let us start from there. Mitochondria, what are some of the features, structures, functions? Cell organelle, when we talk about the mitochondria, what are some of the features we will remember? Some of the features would be, it is double bond. It has got
two membranes, if you see. It is double wall. Now, when I say it is double wall, what do you understand? Which are the two membranes I'm talking about? This and this. Two membranes. And on top of that, the inner membrane is folded. No, this is the inner membrane. Folded and folded and folded. What are these folds known as? The folds are known as cristae. You don't have to remember this word. That's not the inner portion. Cristae. Plural, cristae. Singular, crista. You would say crista. So what are the folds of the inner membrane known as crista? Plural, cristae. And on the inner membrane, this small knob-like structures are there. On the inner membrane, you see this green knob-like, this violet colored knob-like structures, this violet colored knob-like structures. So on the entire surface, there are some small granules as if. So on the inner surface, there are certain granular structures, and these granules are actually the sites where the synthesis of proteins, sites where the synthesis of ATP takes place. We are talking about mitochondria, where the synthesis of ATP takes place. Now, what is the job of mitochondria? What are you supposed to always remember? Mitochondria is also known as that means it must be engaged in the production of energy. Energy in what form? ATP. What is the full form of ATP? Triphosphate. Okay, so this we'll have to remember. Now, when we talk about the features, we already said that the inner membrane, what is cristae, already we discussed just now again. And the cristae has some knob-like structures, and these knobs are the sites where the process of ATP formation or ATP synthesis will take place. Then, if I have to talk about the function, so for every particular organelle, I said how are you going to study? You will always try to remember the structure, certain special features if at all, and the function. Structure, certain particular features if at all, and the function. So when I talk about the function of mitochondria, what would be the function of mitochondria? The function of the mitochondria would be the formation or the production of energy molecule. So you are asked, what is the energy currency produced by the cell? ATP. What is the energy currency produced by mitochondria? Production of energy molecule or energy currency. Production of the energy molecule only I will write in the form of ATP. In the form of ATP. And apart from this also, the mitochondria also has the capacity of production of a variety of other different type of proteins and enzymes, and carry out the process of cellular oxidation. Carry out the process of cellular oxidation. Now, what is the meaning of cellular oxidation? When I talk about cellular oxidation, every particular cell, now that is not to write. If you write, that is just for your understanding. This point you may not write for your exam. These things you will learn in the 10th. But since we are talking about this cellular oxidation, I would love to mention what it is. Cellular oxidation. So when I talk about cellular oxidation, what is the meaning of this? Every cell would generally have what? Food stored in it in the form of glucose. No? And that glucose has to be broken down. So this glucose would get broken down in the presence of oxygen, and then only the carbon dioxide and the water would be released, and also here also energy production happens. So this process, which is constantly happening in our cells, is what is known as cellular oxidation. Okay. So is the mitochondria also helping in the process of cellular oxidation? Yes. Why is the mitochondria? You also get a question here. Why is mitochondria known as the semi-autonomous organelle? Why is the mitochondria known as the semi-autonomous organelle? The mitochondria is known as the semi-autonomous organelle only because it has got its own DNA and it can regulate the process of formation of the enzymes and proteins and its own division. Okay, it can regulate the process of formation of enzymes, proteins, and its own division. Is it fully dependent on the nucleus? No, partially dependent on the nucleus. So this is about the mitochondria. Now we talk about the next organelle. The next organelle is endoplasmic reticulum. Let us talk about this. Endoplasmic reticulum. Check here.
It's written after enzymes
and carry out the process of cellular oxidation. Now endoplasmic reticulum, that's the next organelle. Let us study. And 3 organelles, one after the other, we will check. All of them are more or less related when we talk about. So let us start with the next organelle, endoplasmic reticulum. Endoplasmic reticulum. Okay, we are now studying endoplasmic reticulum. We try to understand there are certain tubular arrangements in the cell like this. You can refer to the diagram that you all have drawn. Inside the cytoplasm, did you draw some tubular structures running from the nuclear membrane to the cell membrane? And this endoplasmic reticulum has got a variety of functions also. This endoplasmic reticulum has variety of functions. So when we are talking about the endoplasmic reticulum, what are these? What are some of their features to remember? So when I talk about types, functions of the endoplasmic reticulum, they are in the form of tubular structures. They are actually a network of tubular structures forming a network. Forming a network between the nuclear membrane, and the cell membrane, So now, when we are talking about the endoplasmic reticulum, what is endoplasmic reticulum? What to write? What is endoplasmic reticulum? Endoplasmic reticulum is a network of tubules. And these network of tubules can be of 2 different types. I'll show you now. They are of 2 different types. But how did we do them? How did we draw them? If you remember, some tubes we had drawn constantly from the nucleus to the cytoplasm, to the cell membrane. Now, whenever we are talking about the endoplasmic reticulum, how to define? A network of tubular structures spanning the entire width of the cell actually, from the nuclear membrane to the cell membrane. Then, what are the other features that we have to write down? They may contain, may or may not have ribosomes attached. May or may not have ribosomes attached. They may or may not have ribosomes attached to them. So there is another property that we understand. Endoplasmic reticulum. Let us talk about their types. Now all organelles may not have types, but endoplasmic reticulum has. So what are the types? We are talking about 2 types. One is known as rough endoplasmic reticulum. One is rough endoplasmic reticulum. Try to understand and follow also at the same time. If at all anything is not clear, ask. So rough endoplasmic reticulum, we can also write in short RER. What is rough endoplasmic reticulum? What will you write? When ribosomes are attached to the membranes, of endoplasmic reticulum, they are known as rough endoplasmic reticulum. Then what about the second? If I talk about smooth endoplasmic reticulum, what is this? When we talk about smooth endoplasmic reticulum, in case of smooth endoplasmic reticulum, also known as, or in short, known as mentioned as SER, do not possess, they do not possess ribosomes attached. They do not have ribosomes attached at all. So the two types of endoplasmic reticulum very nicely you have to remember now. When I talk about the endoplasmic reticulum, what are their types and how do we classify their types? How do we classify them? The classification is mainly based on whether endoplasmic reticulum has ribosomes attached or not. So let us now look at the functions first of all. Endoplasmic reticulum, do they have any other specialized property as such? When we have been talking about mitochondria, we said one specialized property of the mitochondria is the presence of their own DNA. Do endoplasmic reticulum have anything like that? No, they don't have anything. Now that I talk about the types, this endoplasmic reticulum can have two types. Now, if I talk about one type of endoplasmic reticulum, see here, you can very well clearly make out that the outer membrane has got many granular structures attached. And this is actually synthesis of protein. This is actually responsible. This granular structures, what is the name of this granular structure? This itself is an organelle. Which organelle I mentioned already? Ribosomes. And always remember, ribosomes are engaged in the process of synthesis of proteins. But actually, these membranes are not producing proteins. It is the ribosome which produces proteins. So let us talk about the functions. What about the functions of the endoplasmic reticulum? The functions of endoplasmic reticulum, number one would be providing a structural framework. Providing a structural framework to the cell. Now, what do you understand by the concept of structural framework? When we say structural framework, what do you understand? A definite shape, a definite stature, it's not a definite structure when we understand. So a cell would generally have a definite shape and structure also because of the entire arrangement of the tubes inside there. So should we understand this way that if these tubes can be taken out, that means the cell will not have a definite shape. Yes, something like that. The tubes which are arranged give the cell a definite shape. We talk about any type of cell in our body, the skin cells, the muscle cells, the neurons, etc., etc. So any type of a cell you talk about, they have a definite shape because of the endoplasmic reticulum also. And if the endoplasmic reticulum can be removed out from a cell, then perhaps the cell will collapse, perhaps the membranes will collapse. For example, if I ask you that what are some of the parts responsible for the structural framework in our body, then what it should be? Skeletal system. The skeletal system, the bones. So just imagine if your finger doesn't have bones, then you will be sloppy. If your hands doesn't have bones, if your legs doesn't have bones, then our body shape would have been constantly changing like the amoeba. So now when we are talking about a structural framework, a proper shape, a definite shape is always because of certain hard, stiff structures. So the cell also enjoys a particular shape because of this. What else now next is transport. Transport of the proteins, lipids, and other cell secretions. Transport of protein, lipids, and other cell secretions. Now, what is the meaning of that? When I say transport of proteins, lipids, and other cell secretions, say suppose I talk about a plant cell, animal cell. We are talking about animals. This is the nuclear region, and here I have been showing you that if this is the nuclear envelope, then how should the endoplasmic reticulum be arranged? So accordingly now, this is the nuclear envelope, and I am talking about an endoplasmic reticulum which is now present here this way. And this endoplasmic reticulum, say suppose, this endoplasmic reticulum is a rough endoplasmic reticulum. So if it is a rough endoplasmic reticulum, then what do I understand? It should also have some ribosomes attached on its membrane. This is what we have drawn also earlier. It should have some ribosomes attached on its outer membrane. Now, what am I saying? It very well helps in the transport of cell secretions. It also very well helps in the formation of certain type of chemical substances and also its transportation. Okay, let us complete another point to understand this better. Transport of proteins and lipids, and now if I specifically talk about RER, I am talking about function. RER helps in engaged in formation of proteins. Formation of proteins. RER engaged in formation of proteins. What about SER? SER engaged in synthesis of fats, lipids. So, say suppose now I talk about that this is the rough endoplasmic reticulum. So, what is it supposed to produce? It is supposed to produce proteins. It is supposed to produce proteins. So, say suppose inside the endoplasmic reticulum it has been producing the proteins. It has been producing the proteins. So, what will happen? These proteins, the moment they are filled up in this tubule, they will be collected in some of these vacuolar structures, in some small, these are called vesicles or vacuoles because these are nothing but bubble-like structures getting formed, single membrane thin structures. So inside this, the proteins will start getting filled. After some time, what? After some time, they will also start getting detached from the membrane of the endoplasmic reticulum, resulting in the formation of a complete packet of vesicle. What do we call this? Such type of structures are known as vesicles. Such type of structures are known as a vesicle. So what is this vesicle carrying now? Proteins. So it is possible that this may also come and fuse with, the cell and release the proteins out of the cell. Maybe the protein has got some function outside the cell in another cell to be performed. That also is possible. So can I say that the endoplasmic reticulum is helping in the transportation of these proteins? Yes. Something more also can happen. This is one. So what am I trying to tell you? The second function where we had written that endoplasmic reticulum helps in transportation of cell secretions. So now I'm saying that the endoplasmic reticulum is very much helping in the transport of the proteins itself that it produces. It is producing its own proteins and it is helping in the process of transport. But something more also can happen, that very close to the endoplasmic reticulum, it is also possible that there will be another set of organelles, and these organelles are known as the Golgi. There's another set of organelles and these organelles are known as the Golgi. So what will happen? This endoplasmic reticulum, this vesicle will also move towards the Golgi. So two things that are at the same time I'm saying. Can they happen parallelly? Can they happen together? Yes. Some vesicles will move towards the membrane to be released out of the cell. Some vesicles will move towards the Golgi. So now if they are moving towards the Golgi, then what? They will come and get fused with the membrane of the Golgi this way. Quite a number of times, certain proteins which are produced by the endoplasmic reticulum may not be fully functional. The proteins which are produced by the endoplasmic reticulum may not be fully functional, but they are getting matured here inside another organelle. Now, what is the meaning of maturity for a chemical component? When I talk about most of the chemicals which are produced, first of all, if I talk about the enzymes and the proteins produced by our cells, let it be the endoplasmic reticulum, in the muscle cell, in the stomach cell, anyway, right from the time now, right, that they are released from a cell, did they start showing their function? No, they will not be able to show their function. They need little bit of maturity. The chemical substances also need little bit of a change in their composition. For example, like change in the hydrogen and the oxygen and the carbon positions, and then now they become functional. Exactly same like a small baby, right from the baby is born. Can we ask the baby to brush its teeth and eat food? Of course not. So it needs a little bit of maturity and growing up now. So to show a particular function, these proteins has to go through certain modification. So where is this modification done? This modification will be done in the Golgi. So all these enzymes are getting matured in the Golgi. And after that, this vesicle, once it is released from the Golgi, the proteins here and the proteins here have got some changes in their composition. So I would say this is a non-functional protein and this is a functional protein. So we'll talk about the role of Golgi again later, but as of now, for the endoplasmic reticulum, what are we saying? This endoplasmic reticulum, is it not helping in the transport? Yes, it is very much helping in the transport of the cell secretions.
But how will it transfer outside?
It can. Whenever we are talking about, say suppose the muscle cells are producing proteins. A particular cell will produce excess amount of proteins or variety of different type of proteins. And that protein can function within the cell also and it can go to the neighboring cell also. You talk about the stomach cell. The stomach cell is producing the enzyme, but the food is not entering inside the stomach cell. No? The stomach cell is just pouring the enzyme outside in the cavity of the stomach. The food is in the cavity. The enzyme is just dropping as if like showering on the food and then the digestion happening. So these proteins coming from endoplasmic reticulum and some of them may be gathering maturity in the process and then they release. Some will get maturity in the Golgi. So that is what to remember as of now. Synthesis of proteins and lipids, this property you always have to remember. One major difference always you are asked, differences to write between RER, SCR. So this is one. Transport of proteins we already understood. Third, and then the fourth now. What is the fourth? Another function of the endoplasmic reticulum is known as membrane biogenesis. Membrane biogenesis. Now what is the meaning of membrane biogenesis? Membrane biogenesis means formation of new membranes. And what is the composition of the membrane again? What is the composition of the cell membrane again, if I ask you?
It is lipoprotein.
Lipids and proteins. Lipids and proteins. So, if I have to talk about that, the formation of the formation of the membranes in a young cell. Now, this is a cell. Say suppose it divides and it forms two new cells. The nuclear membrane is forming new again. The cell membrane is forming new again. So for the formation of the cell membrane and the nuclear membrane, will it not require a large amount of proteins and lipids? So which organelle is producing the lipids and the proteins? The endoplasmic reticulum. So if in a cell the endoplasmic reticulum is very less, will that cell be effectively and fast able to show division? No, that cell will not be able to show division because the membranes are not getting formed. How can the new cells get formed? So formation of new membranes of a cell, newly formed cell, that is called membrane biogenesis. And the last function is known as detoxification. You know this word? What is the meaning of this?
To detoxify or remove toxins from something.
Toxins. Now, how do you detoxify your body? What is the simplest way by which we try to detoxify our body?
Different sort of diets we follow.
I don't follow any diet. We drink lots of water.
Yeah.
No, that's the simplest way we all do. We drink lots of water to flush our body, run our body of toxins, harmful chemicals, something. That's the simplest thing that we do. Inside our body, there are certain specialized type of cells which are constantly doing this job of converting the harmful chemical substances that are entering our body to less and less and less toxic forms. Now, which are those cells I'm talking about? Now here, if I talk about that RER is synthesizing proteins, one example you can write down. In which type of cells of our body RER is found in large numbers? Something like the muscle cells, if I have to mention, especially the skeletal muscle cells actually. In the skeletal muscle cells, the process of contraction is very, very much extensive and they require so much of proteins. So the muscle cells are one particular site where the RER synthesis is maximum. Then what about SCR?
In what kind of muscle cells?
Skeletal, I said. Muscle cells also you can write, but skeletal would be very, very specific to write down.
Yes.
Skeletal muscle cells. No? 3 types of muscles you have come across ever? Okay, you learn here. So when we talk about the 3 types of muscles, one type of muscle which makes our hands and face and legs, These are attached to bones directly. So that's why we say skeletal muscles. They're skeletal muscles. So in this type of muscle cells, the number of RER is maximum because so much of proteins are required. Every minute, every second, we are contracting these parts of our body. And is SER, if I say, SER is involved in the synthesis of lipids. And which type of cells would generally have this type of ER in large number? That would be the liver cells. The liver cells of our body has got a large concentration of SER. Is it necessary that any cell that I talk about should have equal number of RER and SER? Not necessary, depending on the function. If it is a liver cell, then they will have more and more of SER. If it is a muscle cell, then they will have more and more of RER. Okay. Put off the AC. Where's the remote? Oh, the AC is facing you all. So now, when we are talking about this detoxification, let us learn this function. What is detoxification? So from this word, you easily understand we are talking about breaking down the toxins in our body. Now, how does the toxins in our body get their entry? On a daily basis, does toxins in our body get their entry inside our body? Do we take in toxins in our body? How do we take in toxins in our body? Are the liver cells constantly engaged in breaking down the toxins to less toxic forms? Yes, every day. But in what form? Can you tell me in what form we are taking toxins? Daily basis we are taking in a lot of toxins inside the body. How? You don't take toxins inside your body?
We do,
but how? Now that by
eating particular things.
Variety of food. Now say suppose, now that we talk about variety of food items, all these different type of food items, each of these food items have got some or the other percentage of pesticides in them. So yes, small amount of these are entering. Now it is not seen, we don't understand, but it is going. Excuse me. Now at the same time, say suppose how many of you always love to feed on packed foods? I think all of you. If you're not saying also, I'm sure all of you, no? All of you must be depending yourself on some or the other packed food, and that also on a regular basis. They are almost every day. So if that is what it is, then yes, so much of preservatives are constantly entering your body. So these different type of chemical toxins, chemical substances, the moment you are consuming different type of substances like those packed foods and the tinned and the canned foods, yes, they contain so many different type of toxic substances. On top of that, if you are talking about some variety of medicines, people who are consuming different type of medicines. On top of that, if I'm talking about people who are alcoholic, now these are at the highest level, so much of toxins in their body. So these people, but at the same time, we understand that yes, they are consuming alcohol, but still they are surviving for a long number of years. No, it's not that you consume alcohol or something like that in 1 or 2 weeks your life is gone. No, because we have got such specialized cells in our body which can constantly break down the toxins into less toxic forms and release them out of the body. So which cells are doing this function? The liver cells. And which type of ER is involved in this? The SER. And what is the function called? Detoxification function, which is majorly carried out in the liver cells. We are talking about the ER, and with relation to that, now let us talk about the Golgi. Quite similar. We talk about the Golgi complex now. We talk about the Golgi complex or the Golgi bodies, the Golgi complexes or the Golgi bodies. So what are some of the features, differences, similarities? If I talk about the Golgi bodies, they are a network of tubular structures, or they are not necessarily always tubular. They can be vesicular also. They can be vesicular or tubular structures forming a stack, not a network exactly, forming a stack. See there, these are basically stack-like, or sometimes they are tubular structures also, vesiculars also, or you can also write another word, disc-like, because that's the form that we are drawing here. Disc-like or vesicular or tubular structures forming a stack between the nuclear membrane and the cell membrane. And this Golgi complexes that we understand, The Golgi complexes can synthesize. Okay, this is we are right under the function. Do they have any types? No, they don't have any types. The Golgi complexes don't have any types. But the satellite structures that we are talking about, majorly I talk about the disks. The disk or the satellite structures. The disk or the satellite structures are called the cisterna or the cisternae. Plural is cisternae. What was the fold of the mitochondria known as? What was the fold of the mitochondria known as? See here, this is what I was explaining you just now. I forgot to show you this. From the endoplasmic reticulum, it will pass through the Golgi and then also it can leave the cell. Where the Golgi is helping in the modification of the protein that it brought from the RER. What was it? The fold of the mitochondria, what is it known as? Cristae. And the fold of the mitochondria known as cristae. So I'll just show you the structure of Golgi with reference to this only. The Golgi has got, see here, the Golgi may have tubular structures like this. The Golgi may have vacuolar or vesicular structures. These are all parts of Golgi in different stages. And the Golgi may also have sac-like structures in stacks. And sometimes in the shape of vesicles, sometimes in the shape of tubules. Okay, in the same structure, in the same apparatus, in the same organelle, you can find 2-3 shapes at the same time also. One word to remember, what are these sac-like structures called? If I have to label one of them, then what should I label this as? I should label this as cisterna. Singular, plural, cisternae. Okay. Now, what are their functions? If it is the Golgi, then what are they doing in the cell? What type of a function do they perform? The Golgi, to understand, majorly is synthetic in nature. The Golgi majorly is synthetic in nature. So, what are some of the important functions? Secretes 40 different types of enzymes. 40 different types of enzymes. Secretes 40 different types of enzymes. So many different types of enzymes it produces. Secretes 40 different types of enzymes.
The cell is really small.
So just imagine so many. I mean, there are so many others. When we are talking about the Golgi is producing so many different types of substances. Just try to understand, in our stomach itself and the intestine itself, there are so many different types of enzymes produced. So now that you eat an apple, there's a separate enzyme for digesting that. And there are also different components, and not the entire apple a single enzyme can digest. To talk about eating the roti, that's another enzyme has to do the job. That also partly, only this much, one enzyme, the next part another enzyme, the next part another enzyme. So in digesting one roti, maybe 5 enzymes will be involved. So many enzymes the cells are constantly producing, these organelles are producing. What is the first function of Golgi? We are saying secreting a variety of enzymes. So many different types of enzymes. Now, some enzymes are hydrolytic in nature. Some enzymes are hydrolytic in nature. Hydrolytic. What is the meaning of hydrolytic? Digestive. They can break down. I'm sorry. Some enzymes are digestive in nature. Digestive. They are digestive in nature and vesicles containing such enzymes are known as vesicles.
What's the last word in the first line?
Vesicles. Hydrolytic. 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 Lytic. Some enzymes are hydrolytic, that is digestive in nature, and these vesicles containing such enzymes are known as lysosomes. They are known as lysosomes. They are known as lysosomes. So I can also say that lysosome is a separate cell organelle altogether, but the lysosome is formed from the Golgi. You get statements. Lysosomes are formed from the Golgi. Give reason. Yes, lysosome is nothing but a small vesicle. If you see here, when I talk about that right from the Golgi itself, say suppose here I show you. Now, in this case, whenever I'm talking about that, these are small lysosomes which have been formed. What are they containing? Some enzymes they are containing. But these particular lysosomes, from where did they come? The Golgi is constantly producing this, so many they will be producing. A certain type of a vesicle which the Golgi is producing, but some specialized type of such vesicle will be having enzymes which can dissolve some other membranes, dissolve some other organelles. All enzymes will not do the job, but some enzymes can do that job. If that is the job the enzymes can do, then these are hydrolytic enzymes. And such organelle will be termed as lysosomes. So lysosomes also basically just look like this only. They are nothing very complex. Lysosomes, and one last function of Golgi that we have to write, packaging. Packaging, cell secretions. Packaging, cell secretions. Now, what do you understand by packaging? Packaging means what?
Packing something or getting it ready.
Packing something, getting it ready. Say suppose if I give you one of the simplest examples, say suppose I talk about, tell me,
Putting it together.
Putting it together. Can you give me one example of packaging?
We package stuff to
Which is all scattered, all unorganized.
When we are trying to move houses, we package it.
Okay, we pack small cartons so that everything is organized. Something other example, if I give you would be, say suppose the milk package that we get every day, the small milk bottles or the milk packets. So at one time, if I start, there would be the big farms where the cattle are milked. Then in big cans, the milk collected, then goes to the factories for the packaging, and then from the factories again come to the shops and the dairies, and then so there's a route that they have to follow, and then package, make it organized, and that is how we get those 2 packets and 3 packets every morning. No? So exactly the same thing the Golgi is doing. See here, already I explained you this. If I talk about the endoplasmic reticulum which is producing the protein, it releases its protein in some form which may not be immediately functional, but this may go to the Golgi, get modified, packaged again, now it is ready for function. So if this is the function that the Golgi is doing, then this is what we are talking about as packaging. Many different type of cell secretions, either produced by the Golgi itself, or by the rough endoplasmic reticulum, or by the smooth endoplasmic reticulum. In either of the cases, the Golgi itself is producing so many. No? The production will happen, then the packaging, and then the release. If the production is happening here, then come out from here, then go here, because every particular molecule that the RER and the SER produces majorly has to pass through this. Majorly has to pass through the Golgi. Why? Because of the packaging functions. Since I'm talking about the packaging functions, and I already mentioned about this word, lysosomes. So what are lysosomes? Lysosomes are simple but specialized cell organelle which contains hydrolytic enzymes. So that is the definition. Does it have a single wall or a double wall? You get to see single wall. Simple vesicles these are which contain hydrolytic enzymes.
So I wasn't able to understand the function of hydrolytic enzyme.
I'll explain this, the concept of lysosomes again. What is the meaning of hydrolytic enzymes? Suppose I talk about pepsin, an enzyme of the stomach, for an example. I talk about an enzyme of the stomach. The food enters in the stomach, and this enzyme of the stomach falls on the food, is poured into the food, and the food which is in the complex form starts getting broken down into simpler. So what has happened? This enzyme has broken down complex substances into simpler. So suppose I talk about certain type of a substance or a chemical substance which could break down, dissolve, disintegrate If it is able to do such type of function, then such type of enzymes are known as hydrolytic. It is somewhere dissolving and breaking and disintegrating. Okay. So if that is the function, then we will say hydrolytic enzymes, the digestive enzymes. But digestive, not to always use the word digestion for food.
Yeah.
Here, digestion is meaning it can dissolve, it can break, it can destroy. Okay. Let us learn specifically with lysosomes now. Let us learn specifically with lysosomes. Now, when we talk about lysosomes, what are some of the features? Lysosomes are basically vesicles. Lysosomes are basically vesicles. And what do they contain? Lysosomes are basically vesicles and they would contain hydrolytic enzymes. Vesicles containing hydrolytic enzymes. Digestive enzymes containing hydrolytic enzymes. Okay. And can we write that they are originated or formed from the Golgi? Yes, very much. They are formed from the Golgi itself. They are formed from the Golgi itself. But when we talk about the Golgi, the lysosomes can be small, 2 lysosomes, small. And inside the cell itself, they may fuse together. They may fuse together and now resulting in the formation of a bigger lysosome. Is this possible? Yes, it's possible. Containing a bigger amount of enzyme. Then what are the terms used for such lysosomes? The smaller one. These are known as the smaller lysosomes, primary lysosomes. What about the bigger ones? Secondary lysosomes. So what do you understand about the primary and secondary concept of lysosomes? It is nothing but when I talk about 2 molecules, they can come together, fuse and form a bigger molecule, no? Exactly the same thing. 2 bubble-like structures containing enzymes. They may move separately also in the cell, not necessarily that every time they should fuse. They may move separately, also have small packets. But quite a number of times they can't come together. They fuse. Okay? And when they fuse, obviously they'll become bigger. Now they are called secondary lysosomes. Yeah. Then, what are some of the functions? Lysosomes. Lysosomes, remember the lysosomes are basically not producing anything, but they are actually containing the secretions of the cell. They are actually containing secretions of the cell. Now, what would the lysosomes actually do in our body? Destroy foreign substances. Destroy any foreign substance. Destroy any foreign substances. Destroy any foreign substances entering our cell, entering the cell. Destroy any foreign substance entering the cell. So whenever in our cell a bacteria enters, the lysosomal enzyme destroys them. Inside our cell a pathogen enters, the lysosomal enzyme destroys them. So whenever any unwanted substance, germs, pathogens, etc. enters our cell, the lysosome destroys them. Does it have any other function? Yes. The lysosome is also known as the suicidal bags. The lysosome is also known as the suicidal bags. Now, what is it? Why to be termed as suicidal bags now?
Because if any substance comes in it releases some gases.
No. Why is it known as the suicidal bags? The lysosome can destroy or dissolve the membranes and organelles. Membranes and organelles of a worn out and dead cell of the body. Worn out or a dead cell of the body. So under such situation, it will be known as a suicidal bag. What's the meaning that you understand? What is suicidal bag? See, suppose I talk about a cell which is functional, but after a particular period of time, its functional ability is lost. It has lost its functional ability. So after a particular period of time, what happens? Every cell in our body basically has a lifespan. Every cell in our body basically has a lifespan. Certain cells would live for a few weeks, certain cells would live for a few months, and certain cells also for years. So that means in a year time, couple of years time, they get replaced. But say suppose now that the cell, when the cell is a dead cell, what should happen? I talk about certain cells in our body when they are dead and non-functional. To give an example, very simple, I may talk about the RBC cells. The RBC cells has got a lifespan of just something around 3 months time. A particular RBC gets formed from the bone marrow and can function. You know what is the job of an RBC in our body. So the RBC can function only for a period of some 3 to 4 months, not more than that. Then what will happen to that RBC after that 3 months time? What will happen to that RBC after a period of some 3 to 4 months? Where should it go? This RBC would generally start getting collected by the course of circulation. The dead RBCs always start getting dumped. The dead RBCs always start getting dumped into the liver cells. So by the course of circulation, the dead RBCs will constantly start getting dumped into the liver cells. Then what? In the liver cells, they have to be completely shredded and broken down, dissolved absolutely, because these are dead cells. If they are constantly getting dumped and heaped up, the liver cell will stop performing their regular function. It's not so. So the liver cell is very much undergoing the process of breakdown, because the liver cells also have got a large number of lysosomes in them, and these lysosomes will now start getting released, pouring their hydrolytic enzymes on the nuclear membrane, on the mitochondria, on the cell membrane, on the other parts of the cell, and ultimately what will happen? That RBC that I am talking about is completely dissolved, and the small remnants and the molecules that are formed will all get mixed with the blood, and by the way of excretion it is released out. Almost about 2 lakh RBCs get destroyed every day. Are we aware? Are we aware at all? No, because whenever we talk about these remnants and the dead parts of the cells, and 2 lakh cells, you can understand, so it should be a big amount, no? 2 lakh cells, or at least a handful of membranes should be there. Now, this handful of membranes, if it releases by the way of a waste, then we should be feeding them. No, but we don't even understand, because these lysosomal enzymes can completely dissolve and break them. And just by the way of the waste, they get released out of the body. We don't even understand how many cells are getting formed and how many cells are getting died out. So if that is what the lysosome is doing, when the lysosomal enzymes can kill and destroy and dissolve the old and the worn out cell, will it ever do for a functional cell? Will the lysosomal enzyme ever be poured on a functional cell? No, on a functional cell, the lysosomal enzymes will never be leaking and pouring on the nucleus also, membranes of the mitochondria also, etc., etc. But only when it is dead and worn out and functionless, then only this will happen. Then at that condition, what will be termed the lysosome as? It is known as a suicidal valve. So what is this? We had already talked about the endoplasmic reticulum, the mitochondria, the Golgi, the lysosomes. Let us write one question. Write 2 differences between RER and SER. I'll give you 3 to 4 minutes time. See if you are able to write. You will make a table and write 2 specific differences. Write 2 differences between RER and SER. 2 differences between RER and SER. You will make a table and neatly you will write down. Make a table and neatly write. I'm giving you 5 minutes time. After 5 minutes, I'm going to ask. Your sister is to study here now. Last year, I'm told that you're Nishika, no? Nishika's sister. What is she doing now?
She took PCV this year.
Okay. Where is she?
She went to Poda.
Okay, she went to Poda. No, she was in which school? Ryan, she was in. So you are also following your sister's footsteps.
Yes.
Hoping to. You look quite similar also.
I get that very often.
Anybody more whose siblings were earlier here with us? Yes, also? Okay. Sister, brother? Sister. Okay. She was with CBSE or ICC? She was with ICC. You also? Here only?
Yes, my brother was in.
Which batch?
I think so. Tuesday and Friday.
No, I mean, which year?
I think last year. Last year he was here.
Last year 10th he was.
No, not 10, 9 he was here. Last year.
Last to last year he was in the 9th.
Yes.
CBSE?
Yes.
What was his name?
Amor.
Amor. Okay. He completed 10th from here?
No.
He was not here for 10th?
No.
9. Okay. This is cold drink or water?
Even my sister was here, but I think
Which year?
7 to 8 years.
Oh, when I was young. Are you aware what points to write?
Yes, ma'am.
I asked you two differences between SER and RER to write. What is the first difference you will write? You will write the difference on the structure. First difference you will write on the structure. Ribosome present, ribosome absent. That's the first difference. What is the second difference you will write? On the function. So always remember these are the 3 points only you are studying as per your syllabus. So 1 point on the structure, 1 point on the function.
Yes, ma'am. I do an example as well, other than the
Very good. So generally 1 to 2 mark you will get. A 2 mark question preferably. Got the points to write? Now, we were talking about animal cells, but if I have to draw a plant cell and then try to find out what can be some of the major differences between the plant and the animal cell, all of you finished, no?
Yes, ma'am.
Any doubt, anybody?
No.
For you, homework would be to draw the plant cell. That will remain as homework for you. Huh? Draw the plant cell. That remains as homework for you. Neat, well-labeled diagram you will draw. So since I've drawn the diagram, let us just finish with 1 or 2 just terms to understand. What are some major differences between the plant and the animal cell that you should remember? One difference is the nucleus in case of an animal cell always we draw in the center. The nucleus in case of a plant cell always has to be drawn towards the margins, towards the periphery. Why? Because the central portion of a plant cell is always occupied by a big, large vacuole. That is one difference. Yes, if it is a plant cell, the vacuole will be one big vacuole that should always have, or some 2 or 3 vacuoles they may have. And if it is an animal cell, do they have the vacuole? They may have the vacuole if at all, some 2 or 3 small ones, or sometimes they may not have it also absolutely. That's the second difference. Third difference, what about the other organelles? Do they have all the other organelles? Yes, more or less all. But if it is the plant cell, they may have some additional organelles in the form of plastids. The plastids. 3 types of plastids are there, which I'll discuss next time. So what are the 3 differences that just now we have been coming across? And if it is the plant cell, vacuole present, nucleus moved towards the margin, towards the periphery,
as well as
and plastids. Plastids are never present in the animal cells.
Plastids are used for colors, right? Like
Colors, photosynthesis, and also storing functions. Complete your work, all of you. Draw the diagram of plant cell.
yes.
Okay, see you again.